Hi, everyone, and thank you for joining us today. Welcome to today's The Next Generation End-to-End Solution for EV Research, EV Profiler 2, AutoEV, and Aplo Flow webinar with O&I. Before the webinar begins, we would like to go over some quick house rules for the webinar. I'm sure you've all noticed by now, but you do have a webinar navigation panel on the side of your screen. This contains a few different sections. If you'd like to more clearly see one specific section, you can simply click on the expand icon, which is located in the top right corner of each section. Today's webinar will last about 45 minutes, leaving approximately 15 minutes for Q&A at the end. You are welcome to submit any questions you have throughout the webinar via the chat section, which is located within your panel. However, Colby, Corey, and Sal will only be answering your questions during the Q&A session. If they're unable to get to your questions, then we will forward it to the team and we will write you back with the email address in which you registered with. Within your navigation panel, you'll also find a handout section. Here you can find our company brochure and brochures on the new products we are talking about today. So please feel free to have a look there or on our website located at www.oni.bio. Please note that today's webinar is being recorded. Within the next few days, you will receive a thank you for attending email with a copy of today's webinar to refer back to. In addition, a video recording of today's webinar will also be stored on the resources section of our website. And I'd like to introduce everyone to our presenters for the day. So Colby Janowski, Corey Butler, and Sal Fanale. So Colby is currently an applications development scientist here at O&I. She received her PhD from Vanderbilt University studying cell and developmental biology. Now within her current role, Colby specializes in product and assay development for super resolution microscopy. And today she will be covering our newest product, EV Profiler 2. Corey Butler is currently a software product manager here at O&I. He received his PhD from the University of Bordeaux in bioimaging. With his current role, Corey overlooks and collaborates with software development to implement and roll out new software features to customers. Today, he will be covering O&I's latest software rollout, AutoEV. And lastly, Sal is the hardware product manager here at O&I, and Sal holds a BS in biology and MBA from UCSD. Within his current role, Sal is responsible for leading and overlooking the development and marketing rollout of O&I's hardware products. And today, Sal will be covering O&I's newest automated sample prep and fluidics platform, Aplo Flow. And with that, I will pass the presentation over to Colby to begin the webinar.
Thank you, Carolyn. Okay, so before we dive deep into EV Profiler 2, we're just going to do a brief review of what is single molecule localization microscopy and why you might want to choose it over conventional microscopy techniques such as wide field or confocal microscopy. So on the screen here, you see a wide variety of biological objects that really vary in scale, all the way from things like a mouse brain or a C. elegans, which are these very large 1 millimeter to 1 centimeter scales, all the way down to things like antibodies, which can be about 10 nanometers. Now, conventional microscopy is bound by something called the diffraction limit. And the diffraction limit is an inherent property of light that essentially means if two objects are closer than 200 nanometers, that you won't be able to distinguish them. They will look like the same object. And when we're in this regime of really small biological objects, this becomes a problem because we can't distinguish single particles from each other. So in order to overcome this diffraction limit, we can use something called single molecule localization microscopy, which allows us to take our limit of imaging from about 200 nanometers to about 10 nanometers. So now we're going to be able to resolve objects that are about 10 nanometers, which is giving us a big increase in spatial resolution. Now, let's talk a little bit about how single molecule localization microscopy breaks the diffraction limit. So say we have something like a fluorophore attached to an antibody, which is what this cartoon on the screen is indicating. When this fluorophore actually fluoresces, the signal you get looks something like this, where it's quite a large spot. You can see the center is very bright, and then it might have a darker band followed by another bright band. This fluorescent signature can be approximated into a mathematical function that's more commonly known as the point spread function. We can then approximate the center of this point spread function, so where the peak intensity is, and we can use that peak intensity to localize where this fluorophore approximately sits within this larger spot, giving us a very precise localization of our fluorophore. Now, this is an example that just has one fluorophore in it, but let's look at an example where we have multiple fluorophores to see how we can use this to build up a picture. So here I'm showing you 3 fluorophores that are emitting. Now these are emitting all at the same time, and for single molecule localization microscopy, this would be a problem. Single molecule localization microscopy relies on using different chemical properties to make sure that fluorophores are turning on and off so that only a few fluorophores are on at a time and we can localize them really precisely. So if we take these 3 fluorophores that are on right now and kind of look like a massive blob, and we make it so only one of them is on at a time, we can see here's a single fluorophore. We're going to localize to its peak. Now we have a precise location of that fluorophore. We can then look at our next fluorophore and again localize to that peak. Now we have a precise location of our second fluorophore. And then finally, we can go to our third fluorophore and again localize to its peak. Now we have very precise information about the location of these 3 fluorophores. And you can see that now we have a much better spatial understanding of where those fluorophores sit when we compare it to that previous image that was basically a big blob. And this is how single molecule localization microscopy, also known as SMLM, breaks the diffraction limit. And this is very useful for when we want to look at small biological structures. One of those small biological structures that hopefully everyone here is particularly interested in are extracellular vesicles, also known as EVs. And EVs are small particles that are released by cells into the surrounding bodily fluids, and they have the ability to carry us various cargo, including proteins, DNA, RNA, metabolites, and lipids. They're kind of like a way for cells to communicate between each other, even when they're very far apart. However, EVs can be very heterogeneous. And a lot of current techniques are bulk techniques where you're combining a whole EV population into one bulk mixture, and you're looking at them on a population level. But we know that these particles are heterogeneous. And we can see a really good example of how heterogeneous they are in this big mosaic on the screen. You can see the EVs are very different sizes, and also some of them are slightly different shapes. This is one example of how we can use SMLM to provide better insight into heterogeneous particles. However, SMLM does require a large degree of optimization that often makes it not very friendly to new users. When we're specifically talking about EVs, there's a lot of sample prep that has to go into this. So you need to select the right antibodies and the right fluorophores, because not all fluorophores are capable of this blinking behavior required. The EVs themselves are going to need to be captured down onto a surface, so you need some type of surface chemistry. You need to capture your EVs, and then you're going to need to decide on some other reagents, including their concentrations, how long you're going to incubate them for, and you need to figure out if you can store these reagents, and if you can, for how long. Then, once you've completed that complex sample prep, you have to go in and actually image your sample. And this imaging is very dependent on the parameters you use. And some of the things it depends on are things like your imaging buffer, your laser power. SMLM does require high power lasers. And also, because we've captured these EVs onto the surface, we need to be doing total internal reflection fluorescence microscopy, also known as TIRF. And when you're doing TIRF, you need to select a TIRF angle. So this is just a couple of the things that you need to optimize in order to consistently do SMLM. The other thing that I'm not showing you on this slide is once you've prepped your sample, once you've imaged it, you also need to analyze it. And as most of us that have done microscopy know, analysis is no small task, and this is often very difficult. However, this is where ONI comes in to offer you a full solution EV workflow. So starting with our EV Profiler 2 kit, which is the next generation of our EV Profiler kit, this is going to enable you to reproducibly and robustly produce samples. Then we have our Aplo Flow system, which is going to automate that sample prep for you and enable you to produce a much larger amount of samples. Then we can take the samples over to our Nanoimager, and we've developed the imaging parameters you need to image these EVs that you prepped with the EV Profiler toolkit and the Aplo Flow, so that, again, your data is very reproducible and robust. And then we also have our new AutoEV acquisition app, which is going to allow you to automatically acquire an entire chip, which is 4 samples worth of data, and analyze it as soon as that acquisition is complete, making it very quick for you to get to your results. And this is what we call our sample to answer workflow. So EV Profiler 2 with AutoEV is going to take you from sample to answer very quickly and very robustly. Again, this is our EV Profiler 2 kit, so it's the next generation following from our EV Profiler kit. And this kit has really been designed to adapt to your research needs. As I mentioned previously, we've handled things like your surface prep. We give you advice on how to capture your EVs onto the surface. We've already chosen some common fluorophores and stains that we know work really well for this imaging modality. We give you advice on what imaging parameters you should use and how many fields of view you should collect. And then we also give you a start for your analysis parameters. Now, what's new in EV Profiler 2 is we have a new ultra-low background chip that's going to allow for better sensitivity. We have a new pan-EV membrane stain that's been optimized so that you can get accurate size profiling from your EVs. The EV Profiler 2 is compatible with both surface and cargo detection. So this comes with a permeabilization modality, so you can detect cargo molecules that are in the lumens of your EVs. And we're also offering flexible capture options. So you need to capture your EVs onto that chip. And using the EV Profiler 2, you can do that using either a phosphatidylserine-based capture, which is relatively generic, or you can use a biomarker-based antibody capture where you have an option of the common tetraspan in CD81, CD63, and CD9. This is an example of our new pan-EV detection. So what's really nice about this pan-EV detection modality is it's going to allow you to do things like size your EVs and probe for cargo. However, the pan-EV stain that we're using does not require anything specific in the membrane moieties, which is what you might see if you're used to using a membrane stain like WGA or concavulin A. So regardless of the membrane composition of your EVs, the pan-EV stain will stain them very well, making it ideal for using on many different types of EVs. Now, I'm going to go through an example of how you might use the EV Profiler toolkit to answer some scientific questions here. And to do that, I want to introduce the protein ALEX. So ALG2 interacting protein X, better known as ALEX, is a protein that's implicated in the endosomal sorting complex required for transport, which most people know as the escort complex. ALEX for EVs specifically has been implicated in the recruitment of tetraspanins, and in general, it's a relatively well-known EV cargo protein that is present in many types of EVs. So here we're going to use ALEX as an example of how you can probe for cargo using the EV Profiler toolkit. To do this, we first purified PANC1 EVs from conditioned media using size exclusion columns. We did use the Eyes on Q EV size exclusion columns. And for this experiment in particular, we used fraction 7 that was collected from these columns. So the images that I'm showing you here are an example of how we can see that permeabilization allows for us to detect luminal proteins in these PANC1 EVs. So these images I'm showing you here are a full field of view. So it's a full field of view. This is how much you can capture on the Nanoimager when you take one field of view, which I might also call FOV throughout this presentation. Over on the left-hand side, you can see a sample where no permeabilization has been applied. And very few of these EVs, which are basically each dot here is approximately an EV, are positive for ALEX. However, when we use our permeabilization modality, you can see that many EVs in this field of view are now popping up positive for ALEX. So once you've collected that type of data, we're going to need to analyze it and interpret it a little bit. So our raw data is an example of what I'm showing you on the left. So we have lots of EVs here that are being stained with our pan-EV stain in green, a tetraspanin trio stain in purple/magenta, and then ALEX is going to be in orange throughout all of these images, but it's kind of hard to see when you're looking at a large amount of EVs like this. Once we have that raw data, in order to analyze it further, we need to cluster it, and this is done using our Cody analysis software. So here, each of these examples in grayscale is a cluster. So this would be one clustered EV, this is another, and this is another. Once we've clustered on our data, we can use these clusters to do single EV analysis, which is what I've pulled out here for you on the right. So here we have an example of two different EVs that were clustered. And what we can get on a single EV scale is we can get information like area, diameter, how many localizations of tetraspan and trio are present in an EV. So for example, the EV over here on the left has far more tetraspan and trio localizations than the one over here on the right. And we can also look at the number of ALEX localizations present in an EV. Again, the EV over here on the left has far more ALEX localizations than the one on the right. However, it is important to mention here that when you're using the EV Profiler toolkit, we cannot say that a single localization is the equivalent to a single protein. Something that's important to go through here just when we're talking about our permeabilization modality is that permeabilization does not significantly decrease the number of EVs detected. So when we look at both isotype controls and our ALEX stain samples, regardless of whether permeabilization is used or not, we're detecting about the same amount of EVs per field of view, which is around 5,000. This is important because if we were seeing decreases in the amount of EVs detected when we permeabilized our EVs, that would suggest that the permeabilization is destroying some amount of the EVs and with it destroying precious data. Now in this plot on the right, what I've taken is now we're looking at the EVs on a population level. So we can see that this teal color here is EVs that are stained with the pan-EV stain and the tetraspan and trio. We do have a population that stains with only the pan-EV stain. And then this little orange bar down here, which you can really only see in the permeabilization condition where we stain for ALEX, is our pan-EV with tetraspan and trio with ALEX. So those are the EVs we're interested in. We can represent this data in a slightly different way where we look at the percentages of clusters. So here what we did is each FOV, which again contains about 5,000 EVs, is normalized to itself. So we can say within a field of view, what percentage of those EVs are positive for a given biomarker. And here we can see that we're really only detecting ALEX in permeabilized EVs. So here that pink bar that you can really only see over here is the one that we're interested in. And then over here on the right, we can pull up some examples of single EVs that are positive for ALEX. With this specific imaging presentation, the spot size here is indicating the localization precision, so how precisely we were able to localize a fluorophore. And we can see here that these EVs have a varied distribution of ALEX, but that we also have our nice pan-EV stain, which is going to allow us to size these molecules, and the tetraspan and trio staining, which allows us to comply with MISEP guidelines and confirm that this is in fact an EV. Now we can take a closer look just at this bar over here, these two bars over here. So just our ALEX stain samples that are positive or negative for permeabilization. And if we take a closer look, we can look at just the clusters that are popping up positive for ALEX, which in our unpermeabilized sample is very low. It's about 0.5% on average. But when we permeabilize our sample, we see that that percentage increases to an average of 6, but with a range from about 4 to 8. So we're able to detect relatively low abundant biomarkers here. We can also see that these permeabilized EVs do have ALEX negative and ALEX positive populations, with some examples of our ALEX negative populations over here on the left and the ALEX positive populations over here on the right. The other type of information that we can pull out from this type of experiment is size information. So something that's important to note is on a population level, we don't see a change in diameter with permeabilization. So when we look at this plot over here, regardless of whether we're staining for an isotype control or ALEX, and regardless of whether permeabilization has been applied to the sample, we have a very similar distribution of diameters within our samples. And it is a relatively wide distribution. We do see some EVs that are all the way up to 1,000 nanometers, but this distribution is very similar. So we're not losing EVs of a specific size or biasing ourselves when we permeabilize. Well, let's take a closer look at just our ALEX stain samples that were permeabilized, where we know we're detecting ALEX. And if we do that, we can see that there are slight differences between our ALEX positive and our ALEX negative EVs that we're detecting. On average, the ALEX positive EVs have a slightly larger median diameter. The median diameter goes from about 146 nanometers to about 199 nanometers. However, if you look at this plot, you might see that the tails of these plots are slightly different. suggesting that perhaps ALEX isn't just enriched in larger EVs, but there's a specific size where we see an enrichment of ALEX. We can plot this data in a slightly different manner, where we have a diameter over here on our x-axis and ALEX localizations on our y-axis. And here we can see that it does seem like EVs with a high level of ALEX have a characteristic size. So EVs with large amounts of ALEX seem to be between about 400 and maybe 150 nanometers. And as the EVs get larger, you actually don't see a scaling of the number of ALEX localizations. Again, it's important to note here that a single localization does not correspond to a single protein. So I cannot say that 200 ALEX localizations means there's 200 copies of ALEX within an EV, but we can use trends in localization to interpret those as increases in protein content. So for example, an EV that has 50 ALEX localizations likely has more copies of ALEX present than an EV with 5 ALEX localizations. And this is just an example of how you can use ONI's EV Profiler Toolkit to characterize your EVs further and really get information on a single EV basis. And now with that, we're going to go over to Corey and learn more about AutoEV.
Hey everyone, as Colby mentioned, I'm Corey. I'm the software product manager here at ONI. I'm in charge of the Cody platform, and now I'm really excited to introduce to you guys AutoEV, which is our automated EV acquisition, analysis, and reporting pipeline. So as Colby mentioned and just showed you guys with our EV Profiler Toolkit, single molecule localization microscopy provides a really great way to see subpopulations of your EVs. But she also mentioned that it can be quite difficult to optimize, right? There's a lot of settings like focus, laser power, turf angle, all these kind of things that you need to really kind of be an expert to be able to get great results at. And even once you've got those settings, the acquisitions can take a while, right? We're talking about several minutes per field of view, can be quickly an hour to image an entire chip. So what we've done is we've designed software called AutoEV that makes expert level super resolution imaging and analysis of your EVs accessible to everybody, right? So you don't have to be a single molecule expert to be able to get amazing and reproducible results from your EV Profiler kit. So what we've done is actually we've observed how expert users are using the Nanoimager to do their EV analysis, and we've turned that into a linear workflow here that you can go step by step through, and anybody can run that. So what I'm going to do today is just step you guys through the kind of main steps of AutoEV that allow you to get that kind of expert level single molecule results on your EVs without needing that expert level training. The first step is the experimental setup where you tell AutoEV how you prepared your chip, right? What are your staining parameters? What are your samples? What do they look like? What imaging settings you want to use? Where you want to save your data, that kind of stuff. Then once you're done with those inputs, you don't really have to input any more data. It's all set up front. The second step is system calibrations that makes sure that your system is calibrated and up to date right when you're going to start doing your data acquisition so that your data is of the highest possible quality. The next step is a sample verification, which I'll show you in a little bit, but it's really cool. It lets you answer the question of, is this even worth imaging, right? It helps you understand, are your samples correctly attached and captured and labeled by your EV Profiler kit? And for example, is like a positive control looking like a positive control? Is your negative control looking like a negative control? And should you proceed to do your single molecule imaging? Then you can move on to the automated acquisition and analysis. And this is really the best part. It's a really set it and forget it, press run, and it's going to automatically acquire all the fields of view that you've preset for all 4 lanes of your EV Profiler chip without any user intervention. So you can just walk away. And then the best part is you can come back to your machine and not only do you have your acquisitions done, but the analysis have been performed and you have a summary report that gives you really quick insights into whether or not your samples worked and did your experiment work overall. And I'll dive into each of these in a little bit. So let's start with experimental setup. As I mentioned, this is the place where you're going to tell AutoEV how you prepared your sample. So you'll put in a name for the lane where you put in your sample. So it could be like sample 1, for example, and you'll put in some labeling information. If you're using a default configuration of our EV Profiler kit, we have default settings for those that not only give the names of the different channels, but also different laser powers and imaging frames, right? So you don't need to worry about how many frames should I be acquiring. We're going to give you that information through the defaults. So for example, our pan-EV stain, we recommend imaging for a little bit longer to get a really good coverage of your EV. And so you'll see that in this example, when you select those settings, the pan-EV stain is actually getting 3,000 frames instead of the typical 1,000 that we would recommend for just a standard biomarker. In addition to this, the user is going to select, you're going to enter the title for your experiment, where you want to save your data, into which collaboration on Cody, and which analysis settings you want to use to analyze your data, because AutoEV is going to be automatically analyzing all this data right after you're acquiring it. So once you're done with this experiment setup, we move on to the system calibrations that we've made really, really easy, right? This is it can be a difficult thing to do sometimes, is to calibrate your system, make sure that it's ready to go and that everything is all set for your imaging. And so we've turned these into one-step processes to make sure that everything is really consistent. So all you have to do is drop a bead slide onto your scope, hit run, and the channel mapping procedure will automatically find the focus, optimize the illumination, and perform the channel mapping procedure to make sure that your Nanoimager is ready to do multicolor imaging. Once you're done with that, you swap out your bead slide, put on the EV Profiler kit that you just prepared, drop it onto your Nanoimager, and run our system calibration, which again is a one-click calibration for things that can be quite complicated, like finding the best focus, setting the Z-lock, which is our term for maintaining the focus at a nanometer precision across the entire surface of the EV Profiler chip, and setting illumination settings like the turf angle automatically based on your sample. So all those things that you kind of used to have to, poke around, tweak around a little bit. What is the perfect focus? What is the correct illumination angle? All those things are now automated with a single click. And they're optimized for your system and for the sample that you're imaging. So they'll always be the best ones. The next step is the sample pre-check. This is really great. You click a single button, because you're sensing a theme here probably. Click one button and it's going to take a single image from each of the 4 different lanes of your EV Profiler chip and align them side by side. So you can quickly see, what does your sample look like and is it worth imaging? So a really good example of this is if you have a negative control and a positive control on your chip, you can really quickly see, yes, my staining worked, my capture worked, and in my negative control, I'm not expecting to see anything at all, right? So it gives you a good idea of what the density of EVs are, what your distributions look like, and if your staining worked. The next step is setting up the fields of view that you're going to be imaging for the automated acquisition. And we can do this either by selecting the default fields of view positions that we recommend, kind of in the center or middle of the lane, or you can choose wherever you want in each lane, like near the inlet, near the outlet, near the edges if you want to. You have full control over where the imaging is going to happen and how many fields of view are going to image. So if you have a lane and a sample that's a little bit less dense than some of the other ones, you can increase the number of fields of view in that lane to acquire the same number of total EVs, for example. Then once you've got all that set up, all you have to do is hit run, and your chip will move to the first lane and begin the automated acquisition of all the fields of view across your entire chip, so all 4 lanes of your chip. This is awesome, because all you have to do is hit run and you can walk away, you can go grab a coffee, you can go grab lunch, you can go do something else, you can do more analysis, or you can go prepare your next chip, right? You can walk away from the system and trust that it'll be properly in focus with the right illumination settings and acquiring the data throughout the entire 4-lane chip, right? So you don't have to come back every lane and move it. It's going to automatically acquire all 4 lanes at the same time. And this is the best part. It's at the same time, it's uploading the data to the cloud. So once a single field of view acquisition is done, it's uploading to Cody on our cloud and really exploiting all of the power of Cody's cloud analysis and our EV profiling workflow, which is a one-click EV analysis workflow that talks about all of those settings that Colby was talking about earlier, right? So from our localizations, we move into our clustering, and from the clustering, then we move into our subpopulation counting. All that's kind of automated in Cody. And so while your data is being acquired, it's running that analysis pipeline and then bringing back the relevant information to you while you're still sitting in front of the microscope. So while you're acquiring data, you're able to see how many EVs, so clusters, are being counted per field of view while the next acquisitions are occurring. It's really great. Once your acquisition is done, we've got a few things to help you understand, was it successful? Did it work? is it worth continuing imaging, or should I maybe go back and prepare another chip? Or should I, prepare my next chip with a whole bunch of samples that I want to continue processing? One of the nice things is a mosaic view that appears for each lane that lets you see, like Colby was showing earlier, it lets you get a really quick view of each of the different phenotype subpopulations of your EVs. So single, double, or triple positive. And you can click through these to really get a quick understanding of what do these different subpopulations look like visually, and is that what I'm expecting? Are my analysis settings correct? Is this kind of what I'm expecting for my EVs to be looking like? And then beyond that, once all the acquisition is done and all the analysis is done running on the cloud, you can click Generate Report, and you get this awesome one-page report that gives you all the information you need to know if your acquisition was successful. You'll get the number of EVs per field of view is what you see at the top there, and then you actually can compare across the lanes the biomarker, the subpopulations of the biomarkers, right? So what you see here is a chart similar to what Colby was showing earlier, where now you can see the different subpopulations and you can compare them across samples. So you can see how does sample 1 compare to sample 2 in terms of the number of ALIX positive EVs, for example. And then on the bottom, we have the size distributions, which are really interesting because it's not only the overall size distributions, but it's also the subpopulation size distribution. So you'll get information about what are the size distributions of just my single, my double, and my single positives, my single, double, and triple positive EVs, and you can compare that across your lane. So you can really quickly understand what does your sample look like without having to do any further analysis. And really understand that single EV heterogeneity. So what I've showed you here today is that AutoEV provides an automated, reproducible workflow that gives you great results across your EV profiler chips and saves you a ton of hands-on time, right? So the imaging itself can generally take an hour, and the analysis is probably, overall you're saving probably an afternoon of time here. But wouldn't it be great if you could do the same thing for the sample preparation? And I'll let my colleague Sal talk about that.
All right, thank you, Corey. So Colby did a great job walking us through the science of kind of what our detection and characterization of EVs looks like, and Corey's done an excellent job explaining some of the features and nuances of using our AutoEV software to do acquisition and analysis. But as Corey alluded to, there's a huge piece that's missing from this, right, which is the actual sample preparation itself. So most labs tend to have a specialist, right, who's the one who has the magic voodoo, who can purify particles or build these chips. And our goal with Aplo and all of these products as a family is to simplify the entire process and make it so that any researcher in your lab can produce the same results with the same level of variability, and to make it easier and faster to get to your answers. So with that, I'll kind of go through some of the features of the preparation tools we've built here, and then I'll actually try to go into a little bit of a live demo, show you the software. So the whole point of Aplo, and just to let you know, Aplo is a Greek word that means simple or unencumbered, easy, and that's the theme of all of these tools when they're brought together, right? So higher throughput, less hands-on time actually preparing your samples, faster sample to answer timelines, so basically the turnaround from when you purify a sample or get it to when you actually get the analyzed, characterized results, and then being able to build out modular tools so that you can adjust to fit the differing needs of your labs over time. And 1 week you might be doing one kind of experiment, another week you need to do a different kind of experiment. So we don't want to build a tool that only does a single rail straight kind of workflow. And then of course, the most important part of this is that all of these results are reproducible, so that any researcher doing this at any different site you might have can produce the same answers with the same samples over time. And so we already have the EVP2 kits, which include 3 chips, and we've designed the Aplo Flow system to be able to process all 3 of those chips simultaneously. So that's 12 samples that can be prepared in under 5 hours. 85% less hands-on time, meaning to prepare your chips, it takes about 30 minutes or less of hands-on time. As you get used to the kit and you understand the workflow, it takes very little time to prepare Aplo Flow, but to start, it's 30 minutes or less, and then you can come back and get the prepared chips off the system. Faster sample to answer time for us means from when you put the sample on the system to when you get the answer, the scientific answer, not just do I have these EVs, but what is the population characteristics. And using Aplo Flow with AutoEV together helps us reduce that timeline significantly. And finally, like I mentioned, we maintain all of the unique configurations of the EVP2 kit, even when we're doing it automated. So with that, I'm going to do a little dance, switch between my webcam to a live webcam. I'm actually sitting in the lab at the demo machine here, and I'll switch over to show that as well as our software. One sec. Okay, so everybody should be able to see my screen here now, which is our EV preparation software. So this is a very simple workflow to go through our software. The first thing is when you get to this landing page within Cody, you need to name your experiment. Pretty standard. And then you'll notice that there's a drop down here that asks which kit are you using. And these correspond to the 3 different versions of the EVP2 kit that we sell. So it should be a very simple decision. Hopefully you know which kit you purchased and which kit you're going to run for the day. You would pick that kit. So in this instance, I'm going to pick the PAIN-EV plus tetraspanons with a custom biomarker, since that aligns with kind of the theme that Colby went through with her side of the talk. And then you can see that some of the settings change down here. So in this custom biomarker, you may be looking at a surface protein, or you may be looking at a cargo protein like Colby mentioned. So you make that decision here, and you can see the settings adjust accordingly. So in this instance, we'll just leave it as a cargo protein. And then you would tell the system how many chips you're looking to process. And like I mentioned, you can process up to 3 chips, and you can choose how many you're going to do for this experiment. So for now, I'll just do 1 chip, and we'll walk through. You would name your actual sample. So in this instance, I'll say the first lane of the first chip is a control, and then I'll say maybe I have multiple EV replicates, and I want to name those each individually. So this is essentially one simple experiment where I would have a control and 3 replicates of a sample, and I'd want to run them through the system. Now, maybe you want to have multiple source EVs, and so you could set up a second chip. When you go on to the next step, you'll be able to get multiple chips up here, right? And so the second menu of this screen is the actual prep of the deck itself. And so now you can see it showing me there will be 2 chips I have to prepare on the deck. And then it shows you this custom layout of the block. And that corresponds to this block that's on the system. So we've built a specific reagent holding block to the EVP2 kit to make it a little bit simpler to know where reagents need to go on the deck and in which position they have to go. And so depending on the configuration you do on the first screen, you would get to that configuration here, and it would tell you where to put what volume of sample in which position on the deck, as well as loading up your samples. And then you'll be able to click play and actually run the system. And in this instance, I'm not going to do that because I only have the demo on here and I don't want to burn through all of it. But when the kit is finished, it would jump over to AutoEV, and you can see that some of the names would carry through, and you'd be able to run your experiment here as defined. So with that, I think we'll be able to go to questions.
Wonderful. All right. So thanks so much for the presentation, Colby, Corey, and Sal. We're now going to open the floor for questions, which we will be answering live. So once again, if you have any questions, please just feel free to drop them in the chat box. We do already have some questions that came through. So the first one, I will give this to Colby. And the question was, how do I know that I'm actually looking at EVs?
So that's a really good question. And this is one of the reasons why, specifically for the PAIN-EV detection modality, we've included this tetraspanin trio stain. So I didn't get a chance to really go into this while I was talking, but what the tetraspanin trio stain is it's a combination of antibodies targeted towards CD9, CD63, and CD81. So the tetraspanins that are most commonly found on EVs. And that tetraspanin trio there is really there so that we meet the MISF guidelines, and it provides an additional way of confirming that our molecules are EVs. The PAIN-EV stain is specific to membrane, So it will stain any like membrane bound particles. So the tetraspan and trio stain is really there to make sure that you have confidence that what you captured and stained with the Pan-EV stain are true EVs.
Great, thank you, Colby. Our next question I will give to Corey. So we had Ellen ask, our system is isolated from the web. Can your system be set up to delay upload to Kodi?
Yeah, great question. So today, currently, AutoEV requires an online laptop that's connected to your Nanoimager. So it's got to have an active internet connection, but we're working really hard on making it so that it will work for systems that aren't actively connected to the internet. We know that that's a lot of people that don't, in their labs don't have access to internet on their Nanoimager laptop. And so we'll have some stuff to share later in the year about how you can run AutoEV on those machines. So stay tuned.
Wonderful. We had Evangela ask, and I'll give this question to Colby, what cargo proteins can we look at?
So the EV Profiler Toolkit's designed to be very modular and adaptable. So you can probe for whatever cargo proteins you can think of as long as there's an antibody that's available to them. I was going to say commercially available, but that's not even true. You could generate your own antibodies if you really wanted to. So as long as it's a molecule that there's a way to probe for and attach a D-storm compatible fluor for, like Alexa Fluor 647 to, go wild. Whatever your wildest dreams are, you are capable of probing for it.
All right. Question for Sal. Sal, can you comment on how long the Aplo Flow takes to run?
Yeah, so to run 3 full chips takes about 4.5 hours. It depends a bit on which one of the workflows you choose because there's different pipetting steps depending on the complexity of those different workflows. But the chips finish in sequential order. So you can pick up the first chip off the system somewhere around at 3.5 hour mark. And then the second and third chips finish sequentially after that. I didn't really show it. There's a menu in the software when the system is actually running that shows you a live view of what it's performing per chip with a time to completion. So if the system is running and you're curious when you can get the first chip to start imaging, you can look at that menu, see the time it expects the chip to finish, and then come back and pick up that chip. And then you can also start the AutoEV acquisition simultaneously. So if the system is still prepping the second and the third chip, you can pull the first chip out, put it on the Nanoimager, start AutoEV while it's still finishing the preparation of the second 2 chips.
All right, we have another question for you, Sal. Can I run multiple different protocols per lane on the Aplo Flow?
So at the moment, right now, with the first release of the software, all of the lanes of the chip need to run the same protocol. Soon, within the next 2 months, we'll release continuous updates for this because this is all part of the Kodi cloud. And so Corey and I are working pretty rapidly with the software team to introduce more and more complexity so that you'd be able to do different types of workflows on a per lane and per chip basis to really up the complexity you can run in a single run.
Okay, this is from Evangela again. We might have to have a few of you answer this. She said, thanks so much. Last question, please. What lasers does the ONI have? What dyes would be compatible with it?
Yeah, so I can take most of that. I can take that question. So the ONI Nanoimager comes with 4 laser lines. It's kind of your typical standard laser configurations you see on conventional microscopes. So there's a 405 nanometer laser, a 48 nanometer laser, a 561 nanometer laser, and a 640 nanometer laser. So there's a lot of dyes that will be compatible with those laser lines, but what's really important here is the dyes have to be what is commonly called D-storm compatible or SMLM compatible. You might see either of those terminologies. Within ONI's system, the only channel that we really recommend that people put their custom markers in is the 640 channel. So you're using 647 spectrum dyes. And that's because those dyes are very, very reliable for single molecule localization microscopy. The most common dye and one of the best dyes out there is going to be Alexa Fluor 647, which is really easy to find. You can buy it from Thermo Fisher or any other similar large scientific distributor. And it has a lot of different ways you can attach that dye to your molecule of interest. So our general recommendation would be that people start with Alexa Fluor 647.
Great, thank you, Colby. I do see a few people asking about pricing for the Aplo Flow. We're not going to be quoting any pricing on the call, but we can email you directly with the quote. Next question, can you capture concentration with the EV Profiler kits? So that question's for Colby.
So you cannot capture concentration as a, like you for sure have like 10 to the 12 EVs per mil in your sample. You cannot capture concentration like that using the EV Profiler kit like you might be able to with a dynamic light scattering system. But what you can do is you can very accurately perform concentration titrations. So if you start with a sample that you know is 1x, if you dilute that 1 to 10 or 1 to 100, you will be able to see those changes in concentration on your EV chip. And that will be represented as in a change of the number of EVs that are captured onto the chip surface. So you can do concentration titrations, and those will be relatively accurate. They will follow the titration you do, but you cannot get pure concentration information like how many EVs per mil or microliter you have.
Great. We have a question for Corey. Can I use the analysis workflow I've already made in Cody?
Yeah, absolutely. So if you're already a Nanoimager customer and you've been using the EV Profiler Kit before, or if you get your hands on the EV Profiler Kit 2 and you run it manually, let's say in NimOS, or you already have your settings all set up for your analysis the way that you like it, you can select those in AutoEV at the beginning. So AutoEV will, by default, provide some standard settings that should work pretty well out of the box. But if you have your custom settings, you can select those and have all the analysis be performed with those specific settings so that you know that you get the most accurate representation of your sample in the reporting at the end. And if you run it with the default settings, it's not a big deal. You can always just batch rerun all those acquisitions that you just did with your custom settings afterwards if you need to.
Great, thank you, Corey. Question for Sal. We had Moana ask, is your Aplo Flow system compatible with third-party chips?
Because all of this is fully automated end to end, you can only use the O&I pre-configured chips right now to run the kits. We'll have new products being released on our roadmap throughout this year and next that will expand capabilities and new workflows. That's all going to be based on the same labware. So part of building the reliability is ensuring that the labware is also reliable, right? Especially with the automation here. We're pipetting very small volumes, sometimes single-digit microliters. We really can't risk the labware not being exactly the right geometry and configuration in order to ensure that we can reliably run automated samples the entire process.
Great, thank you for that, Sal. Question for maybe Corey and Colby. Can I look at EV morphologies?
So I think that is a question that we can best tackle. So depends on what you mean by look at. So if you would like to visually look at your EV morphology and be like, oh, some of these EVs have like slightly different shapes. Yes, the pan-EV stain, which is again specific for the membrane, is going to allow you to visually see those differences. However, if you're talking more about on the analysis side, if we provide any kind of morphological readout, Corey can explain that a little bit more.
Yeah, sure. So what we do in Cody is we do provide a size of your cluster, so for your EVs, as well as like a shape metric that's called circularity. So how close, how circular are they basically? And with those two metrics, you can pretty much assess obviously the size, so the diameter of your EVs, as well as like how round they are, right? So those are the morphological parameters that we're able to extract today with Cody.
Great, thank you so much, Colby and Corey. All right, let's take a look at the next question. Someone said, I've tried to prepare my own EV slides in the past and they have proven very variable. How has this been addressed?
So when we've gone through this process of creating and optimizing and verifying the EV Profiler toolkit, we spent a lot of time with the surface chemistry to make sure that this wasn't a problem, that our surfaces were very robust and that they were reproducible time and time again. And so we did some development work up front to kind of see how we could make this process as good as we could. And then we also just did these experiments over and over and over and over again. I think over the course of the development of this kit, we used something like 15,000 surfaces to make sure that these results really were as robust as we could make them. So the simple answer is we did it a lot, and when we saw issues, we tried to just address them as best as we could.
Great, thanks for that, Colby. Next question, do any of our tetraspanin antibodies interact with mouse?
So we can only guarantee that our antibodies will react with human proteins. And this is specifically for the detection and capture antibodies we offer. You're of course more than welcome to try and see if they'll cross-react with your mouse EVs. We just can't guarantee it. However, that is why we offer modalities such as the phosphatidylserine-based capture, which is generic. So that doesn't matter what source of EV, like what species your EVs come from. As long as they have phosphatidylserine on their surface, they can be captured by that modality, as well as the pan-EV stain, which is generic for just lipid bilayers. So it's an EV, they have a lipid bilayer. So the pan-EV stain will be able to detect EVs of any type.
Great, thanks for that, Colby. All right, we had someone else ask about pricing again, so we'll just send an email with a quotation on the side. And then we had Ellen ask, does the surface chemistry have preferred buffer conditions or any buffer conditions you shouldn't use?
So all of the buffers come with the kit, and so everything's kind of been optimized to work together. All surfaces are a little bit different, so we would recommend that you stick to using the buffers in the kit, which Sal had a lovely example of in his hands, and that will give you the best results.
Great, thanks for that, Colby. Question for Corey, does AutoEV work with my own surface?
Yeah, so it's kind of the same answer that Sal just mentioned, to make sure that all the automation for AutoEV, so that we're in the right lanes and you have your different samples and all that stuff, to make sure that that works as best as possible, where it's only working with the ONI provided by OR, so the 4-lane assay chip. With that, then obviously you get a lot of advantages, like being able to position very specifically in the lanes where it's very reproducible across samples, and I think that's a huge benefit. So it currently only works with your EV profiler chip. Oops.
Great. Next question is for Sal. Someone asked, can you verify how many chips I can run on the Aplo Flow again?
So you can run any combination essentially of 1, 2, or 3. And you can choose how many lanes you run per chip. So even if you want to do, say, 2 lanes on the first chip, 4 lanes on the second, that would be compatible. And those settings will transfer straight to AutoEV, so you don't have to double up on the data entry side.
Great, thank you, Sal. Someone just saying with you, Sal, someone asked, what else can I run with the Aplo Flow?
The first automation we're releasing is strictly for EVP2 kits right now, and the 3 different configurations of it. And then shortly throughout the rest of this year, stay tuned, and we'll have more automation coming as we release new products.
Great, thank you, Sal.
It's going to be a full platform of automation and kits that produce end-to-end different workflows.
Great. So this question is for Colby. We had one of the labs say that they are looking into buying the kits, but they won't be able to play around with EV concentrations and optimizing protocols. Do you have a set protocol that we could use as a guideline?
Yeah, so that's a great question. And we do have a set protocol. So we have a recommended range of what your EV concentration should be going into the chip. And then the protocol in terms of how much do I add of each of these reagents is very set. You get exact volumes for everything. So really all you have to do is follow along. It's very similar to if you've used something like a QIAGEN mini prep kit, where there's just tubes and you just pipette in and out of the tubes and you don't really think about it. The EV Profiler 2 protocol is very similar to that.
Great, Colby, just staying with you as well. Going back to the one question that was asked before, I believe about the buffer conditions, and then just adding on to the way you answered it, someone asked, how about what the EVs are isolated in?
So this is actually going to depend a little bit on what you would like to use to capture your EVs. So I mentioned we have that phosphatidylserine-based capture. That capture modality is calcium dependent. So if you would like to use that capture modality specifically, we do recommend you elute your EVs or isolate them in a buffer that contains calcium of some kind. If you, for some reason, don't want to do that, or you already have an EV sample that you've already isolated, we do offer a basically concentrated additive that you add about a microliter to your EV sample to boost that calcium concentration to make it compatible with that capture type. So that's probably the biggest thing, is phosphatidylserine needs calcium in there somewhere. But other than that, if you're just looking at something like our tetraspanin-based capture, something like PBS is just fine. So whatever you typically use to isolate your EVs should work just fine.
Great. Question for maybe a combination of Sal and Corey. Someone said, can I run Cody acquisition at the same time I'm running Aplo Flow? Also, can I use my Nanoimager at the same time?
Yes. The answer is yes. So you can, the system is designed, and there's, like I said, there are prompts in the software interface to tell you when it's safe to take a chip that's been finished processing. And within Cody, you can open up AutoEV at the same time while Aplo Flow system is still running, and you can kick off the second chip acquisition and AutoEV process.
Wonderful. All right, and that looks like all the questions that came through. And it seems we're getting very close to time. So I'd like to thank again to Colby, Corey, and Sal for the presentation. If you have any more questions that come through, we will email them to our team to answer. And once again, the webinar was recorded. Therefore, if you need to refer back to it, you can. All these recordings can be found within the resources section of our website, so make sure to check that out. And thank you to all the attendees for joining us today. Upon exiting the webinar, you will receive a survey. Please fill out the survey and provide us feedback on your experience today, or tell us if you're in need of being contacted directly by ONI. I know some of you did ask for the quote request in the chat, so we'll make sure to forward those on to the sales team to get you a quotation. So thank you again, and we hope everyone has a wonderful day.